Peter Mindek, TUV, multi-scale procedural animations of microtubule dynamics based on measured data. Life of cells is carried out by molecular processes. One of the challenges in the study of these processes is that they cannot be seen. Molecules can be observed with electron microscopes, but in order to do that, they need to be frozen. So while biologists can learn a lot about molecular processes through various experiments, they remain hidden from their view. This is an electron microscope image of a virus. We can resolve the outer membrane with its embedded proteins, and we can recognize the capsid, the structure inside which holds the genetic information. Well, that's already amazing. As you can see, the resolution is quite poor. And more importantly, we cannot observe the dynamic processes which exist within the virus. What we can do is to use computer graphics to model and display the virus on much more detailed levels so that the individual molecules are visible. With the proper means of modeling dynamic processes, we could show them as well as molecular animations. A mechanism for authoring such animations is the core contribution of our work. To demonstrate our technique, we choose a dynamic process which occurs in all eukaryotic cells, the dynamic instability of microtubules. Microtubules are filaments which constantly rearrange by growing and shrinking. We can see the process in this sequence where biologists were able to highlight the growing ends of the individual microtubules. Dynamic instability is intensively studied. Biologists are particularly interested in the exact mechanics of how the microtubules assemble and disassemble. They are long hollow polymers made of protein called tubulin. We have some understanding of how the tubulin molecules bind together to make the microtubules grow and then disassemble again. But we are still learning new aspects of this process as we cannot directly observe it under a microscope on the molecular level. To model this dynamic process, we introduce multi-scale procedural animations. Typically, animations are made by creating keyframes. Here we can see an example of a process where scattered molecules form a sphere. To create an animation of this process, we can model the initial state, the molecule scattered in space, and the final state, the sphere made of molecules. By interpolating these two keyframes, we get an animation. However, this is not how things work in biology. When assembling into large structures, molecules have a precise sequence in which they join the structure. Therefore, we introduce the concept of instance-dependent timing. With this mechanism, we are able to assign each individual molecule a time window in which its interpolation between the keyframes will occur. Another issue is that the molecules don't directly fly to their destination. They are driven by random Brownian motion, and they follow various chemical pathways before becoming building blocks of the assembled structure. To model such behavior, we introduce another concept, instance-dependent transition function. This mechanism allows us to procedurally define a spatial curve along which the molecules will move while interpolating between the keyframes. These curves are parameterized by the instance ID, and therefore they can vary between individual molecular instances. Finally, we introduce time-varying keyframes. While traditional keyframes are simply describing the position and orientation of every item in the scene, we specify keyframes as functions. For instance, to model a molecule's diffusing through Brownian motion, a keyframe would be a continuous noise function where the instance ID would serve as a random seed. Such keyframe would represent molecules randomly moving through space. If we wanted to transform them into a sphere which rotates around y-axis, our target keyframe would be a function which procedurally describes the rotating sphere. Now we can at any point interpolate between these two functions, utilizing the instance-dependent timing and instance-dependent transition functions. This will result in an animation of a procedurally defined dynamic molecular process. To demonstrate these concepts, we implemented a pipeline for visualizing dynamic behavior of microtubules. The pipeline consists of 4 stages. A fiber generator generates the network of microtubules in a cell. Each filament is represented by a spline defined by several control points. Microtubule generators generate a molecular model of the assembled microtubule along each curve. Cap generator generates the growing end of a microtubule. There's a specific way of how tubulin molecules assemble, and this is visible in the specific shape of the microtubule's end, also called a cap. And finally, we render the models with a real-time molecular renderer. The pipeline allows us to model the dynamic instability and easily change all the relevant parameters, making it a powerful learning and hypothesis generation tool. Let's take a look on how these dynamic instability animations created with our method look like. Here we can see the assembly of a microtubule. The randomly scattered molecules of tubulin form a flat sheet which closes into a hollow tube. This is one of the theories of how the microtubules assemble. Here we can see the process of the microtubule disassembly. It starts with the rows of individual tubulin molecules called protofilaments to split apart. This event is called a catastrophe, and this is when the microtubule growth stops and it starts to fall apart. The disassembly stops in an event called a rescue, and the microtubule starts to grow again. Our goal is to one day have a model of an entire cell, not just a structural model, but also a functional one. While there has been a lot of work done on representing and rendering large static molecular structures, we are still missing means to represent the processes. This work is a step towards having a robust framework for authoring and representing molecular processes. Thank you for your attention.